The overall goal of this procedure is to generate adult mice from induced pluripotent stem cells, or iPSCs, by tetraploid embryo complementation. This is accomplished by first deriving iPSCs from mouse embryonic fibroblasts using lentiviruses carrying OCT4, SOX2, KLF4, and c-MYC, and supplementing the media with valproic acid, or VPA. Once the iPSCs are generated, they are characterized by immunostaining, chromosome counting, and embryoid body formation. The next step is to generate recipient tetraploid blastocysts via electrofusion and in vitro culture. Subsequently, the iPSCs are injected into tetraploid blastocysts, and complemented blastocysts are transplanted to recipient females for gestation. In the final step, full-term animals are delivered by cesarean section and fostered to surrogate mothers so postnatal development can be monitored. Ultimately, if viable full-term animals are generated, the assayed iPSC line is considered fully pluripotent.
The main advantage of this technique over other methods of generating iPSCs is that our method reproducibly generates fully pluripotent iPS cell lines.
iPS cell lines generated by these methods may be useful to other investigators wishing to compare the pluripotency of their iPS cells or to establish the equivalence of other reprogramming methods.
These methods will be useful for researchers wishing to generate fully pluripotent iPS cell lines for specific downstream applications such as in vitro differentiation assays. Further, the generation of all iPSC mice using tetraploid complementation may serve as a useful means to determine the genetic and epigenetic stability of various cell lines.
The micromanipulations involved in tetraploid embryo complementation are technically challenging. It may take several months for someone to become proficient at the techniques.